everyone, and welcome to today's webinar, Mastering Multiplexing, Innovative Tools for Multiplexing in Your qPCR Workflow. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, we encourage you to participate today by submitting as many questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you're experiencing any technical issues, please use that Q&A box as well, and we will answer you directly. I would now like to welcome our speaker, Dr. Chia-An Yen, Molecular Biology Scientist 3 at Thermo Fisher Scientific. For a complete biography on Dr. Yen, please visit the Biography tab at the top of your screen. Dr. Yen, welcome. You may now begin your presentation.
Hi, everyone. Thank you for joining us today. I'm excited to talk to you about multiplexing and qPCR. This is a subject that is of great importance to the progress of research and development in many different fields, including that of the private and nonprofit sectors, and particularly important for molecular diagnostics and biopharma R&D fields. In today's webinar, we'll start with a general explanation of what multiplexing means in qPCR, followed by why one might consider using multiplexing in qPCR. Then we'll go into the key factors that influence the success of multiplexing in qPCR. What is our goal here? We want to help equip you with the understanding that you need to successfully use multiplexing to your advantage in your workflow. Throughout this presentation, we'll also show you data that can shed light on the importance of using specially designed reagents for multiplexing and how that can be a great ally in helping you achieve reliable and dependable results when doing multiplexing with qPCR. So what exactly does multiplexing mean in qPCR? Multiplexing in qPCR allows you to amplify more than one target in a single qPCR reaction within a well. How is this achieved, you might ask? It's all about using different reporter dyes, each with a unique fluorescence spectrum that can be distinguished for generating different signal output with the qPCR machine. Let's take the example on this slide where I have a multiplex reaction of 4 different targets. So for each of these targets, they have a assay, so the primer and probe sequence that target the region of interest. For the probes used in this case, target 1, 2, 3, and 4, they each have an assay with a unique reporter dye. If you mix all of them together with master mix and template or your sample, run the qPCR reaction, you end up with 4 distinct signal output, one for each of the different target. Now let's turn our attention to a different multiplexing scenario where you might not care about distinguishing the signal coming from 2 different targets. So in this case, we have 2 assays, one targeting target 1 and 2, and assay 2 has target 3 and 4. So here for assay 1, I will have probe sequence for target 1 and 2 that are different, but the reporter dye used is the same. And that way, the signal generated will be combined as a single amplification curve. And that can be a use case scenario. For example, if you're trying to detect different targets of a virus to make sure you can catch the presence of that virus without an issue and with more confidence, you might want to combine two targets targeting the same virus for assay 1, and maybe you have target 3 and 4 targeting a different virus of interest. You mix them together with master mix and template, run the qPCR, you end up with two distinct amplification curves, one for target 1 and 2, and one for target 3 and 4. Now that we've gained a better understanding of how multiplexing in qPCR works, it's time to go into the rationale for consider this approach. So what are the advantages in multiplexing in qPCR? Imagine you're working with samples that have low yield. Samples with low yield might not be sufficient for amplifying multiple targets in separate reactions. So what's the solution, you might ask? Multiplex in qPCR. So say you have 10 samples and you need to amplify 5 different targets for each of the sample. With multiplexing, you would only need to run 10 reactions, one for each sample, versus 50 single target reactions if you don't do multiplexing. As this demonstrates, with multiplexing, you can significantly reduce the amount of sample you need. But the benefits do not stop there. Less sample means less reagent, and less reagent translates to lower costs. As for the benefits for your workflow, multiplexing can help streamline it and reduce the time needed to complete your work, as well as increase the throughput. And finally, let's not forget that with multiplexing, you're getting less variation because comparing the different targets in the same reaction is better than comparing them across different reactions. As you can imagine, there are different factors that can impact how variable the results will be. And now that we've seen the advantages of multiplexing in qPCR, let's shift our focus to understanding the factors that can impact its effectiveness. So how many of you have faced challenges with multiplexing in your work, specifically for qPCR? I'm gonna guess that this is not something that's unique to a small group of people. As we know, multiplexing is a complex process. So diving into the challenges with multiplexing in qPCR, first of all, the application itself can be complicated. So say if you're looking at a gene expression profiling with your multiplexing in qPCR, you will need to make sure that you can balance the amplification of the different targets, which may be present in varying level in your sample. And again, that will depend on your sample type. And for the case of pathogen detection, where you really need a good sensitivity and specificity, that can be complicated with multiplexing, where if there's interactions between the assays, you might not be able to amplify your pathogen with confidence. And so specificity might suffer, or if you have interaction between the assays, you can also have reduced sensitivity. So sample type adds another whole layer of complexity. So sometimes you might work with challenging samples or something that's really low yield where achieving a reliable result is really difficult. So for the next item on this list, assay design. This involves the selection of sequences for both primer and probes, as well as the choice of reporter dye and quencher for the probe part. So all of these different pieces can interact between each other and make the amplification of a target more of an issue versus the other. So we'll dive more into this later, but just, keep this in mind. Assay design is perhaps the most important thing affecting your multiplexing success. And next, master mix compatibility. So not all master mix are made the same. A master mix that's developed for multiplexing will definitely have an easier time amplifying multiple targets in a single reaction compared to one that isn't designed for it. And then next, thermal cycling conditions. So this is where after you have your assay designed, you may need to adjust the thermal cycling protocol to find the sweet spot that can be the most specific and most sensitive to all your targets in the multiplex reaction. And then lastly, let's not forget the real-time PCR instrument and dye compatibility. So this is perhaps one of the first thing you want to look at because you want to make sure that the instrument you have the proper excitation and emission filters that can allow you to detect the reporter dye that you want to use in your assay design. So all in all, multiplexing in qPCR isn't a walk in the park, and there are many different factors to consider. Now that we've laid out the key considerations for multiplexing in qPCR, let's go into the assay design part. As we mentioned, this is, the most important piece for the multiplexing success. So once you know the dye compatibility of your real-time qPCR instrument, then you know which dye you can use for the different targets in your multiplexing reaction. So here I'm showing you the 6 different reporter dye that TaqMan probes have that you can order through Thermo Fisher's website. So here we have Fenvic Abbey June, signing 5 and signing 5.5. So all of these reporter dyes, they each have a unique emission spectrum that allow them to be separated by different filters. As you can see their wavelength on the right. With these different reporter dyes, you will want to be mindful about what quencher you pair with them. So for Fenvic, Abbey, and June, we recommend pairing them with the MGB or QSY quencher. And just as a note, if you have one of these reporter dye in a specific quencher that's already used, if you're trying to switch between MGB and QSY, you will definitely need a probe redesign due to the change in TM. And then so next we have signing 5 and signing 5.5. These two reporter dyes, we recommend pairing them with QSY2 quencher, and this is recently available on our Thermo Fisher website as well to facilitate your multiplexing need. So let's talk very briefly about, how would you want to assign the different targets to specific reporter dyes? And here is a very general rule for you. So roughly you want to pair the brightest dye with the lowest expressors in the multiplex reaction and vice versa. You want to pair the dimmest dye with something that's of stronger signal or more highly expressed. So for our reporter dyes here, Fram and Abbey are the brightest. So you would pair them with something that's lowest expressed in your multiplex reaction, followed by Vic in June. Cyanine 5 and cyanine 5.5 are the dimmest dyes, so you would definitely want to pair them with something that's more highly expressed or perhaps the internal positive control target. So designing an assay for multiplexing is like solving a complex puzzle where numerous factors can impact the reliability of your results. So one of which is the nonspecific amplification in your null template control reaction. So this could arise from increased interaction between different assay sequences and or templates within a single reaction. Then we touched a little bit about the reporter dye and quenchers. So specific pairings may help minimize background and limit crosstalk between targets. And then next, there's the thermocycling condition, which you would want to make sure that you have all your assay design with close enough an optimal annealing temperature in the multiplex reaction. So competition and inhibition between assays is another huge factor, and that can occur through interactions among various primer pairs, probes, targets, amplicons, or any combination of the above. And you might want to check for that with running a single target reaction for all the different targets and compare them to the multiplexed reaction. We've also touched the importance of pairing dyes that are brighter or dimmer with targets that are lower or higher expressed. Another aspect of this is you can also vary the amount of primer and probe in your assays depending on the expression level of your different targets. And finally, let's not forget the efficiency of qPCR reactions. You want to make sure that all your targets in the multiplex reaction is amplifying at optimal efficiency. And this can be especially of an issue in a multiplex reaction where they can be interactions between the different assays and targets. For more detailed information regarding multiplexing and assay design, we do have our Tagman assay multiplex PCR optimization guideline online that you can definitely sign a request for links with this webinar to go and check it out on your own, as I don't have the time to cover all the information in there today in this webinar. So with all that being said, we know that assay design can be a complex process, and that's why we've got your back. So on our Thermo Fisher website, you can find pre-designed assays for various applications and species. And so these assays are ready to use. They're guaranteed to perform in your real-time PCR and digital PCR instruments. And these assays are also verified using up-to-date annotations in our gold standard Tagman assay chemistry. But what if you need to design your own assay? So we have the Assay Design Hub on our website that have self-service tools that guide you to different application type depending on what you try to design. And then you can submit a order for these custom assays with the different reporter dyes and quenchers. And if both of these options are out there for helping you with getting successful result with your qPCR experiments, and we're here for you. If you need help with multiplexing, say for the per design assay, you can send a request through our assay design hub to have someone assist you with whether or not you can put certain assays together. So whether you've done this before and you can do it on your own, you can use the custom assay side, or if you're just getting started and you need help, we're here to help ensure that you can do this with the highest chance of getting optimal results. Now that we've covered the basics of assay design and how we can support you in that process, let's go into some data on how using the right tool for higher order multiplexing can affect the success of your qPCR experiments. So when it comes to multiplexing, starting with reagents specifically developed for multiplexing applications can make a world of difference. Take, for example, our latest addition to the Tagman custom assay offering, the QSY2 probes. So these QSY2 quenchers are for, again, stannine 5 and stannine 5.5 dyes. And these probes are designed for higher order multiplexing where you can multiplex up to 6 targets in a single reaction by using the 6 reporter dyes we have. What makes these guys stand out? They have high sensitivity, good dynamic range, and shows no change in performance when you scale up from single target to multi-target reaction, as you'll see in the following slides. So multiplexing can sometimes lead to a loss of sensitivity for different targets due to competitive inhibition. So here we have a comparison between a 6-plex reaction run with our Tagman QSY2 probe compared to probe from another supplier. So here I'm graphing the difference in CT between the supplier probe and our probe. So a higher positive bar means later CT from the supplier, and a negative bar indicates later CT from our probe. So as you can see, with different sample input and the different targets in the 6-plex reaction with the different colors, our probe consistently perform better than the supplier, as indicated by the positive bar graphs showing earlier CQ. And this is particularly important where you check the lowest input. You can see that with the supplier probe, there is a lot later CT, indicating loss of sensitivity. And that on the right here, you can see the amplification path for the target. That's for dye number 6 in gray here, where there's no change in performance as indicated in our graph on the left. But if you look at the end plot, you can see that the amplification curve are definitely less consistent and shows a flaring. And there is also a drop in delta Rn, as well as a loss in detection in the 0.1 nanogram input. In essence, this data demonstrate that with our Tagman QSY2 probe, you can have better sensitivity and earlier CQ in multiplexing reactions. So for multiplexing, a good dynamic range is key. Why? Because it allows for accurate detection of all different targets in the sample, which might be present in various amount of quantities. So let's check the dynamic range for our QSY2 probe in red compared to probes from supplier in blue. So here I have a 6-plex reaction with our probe. You can see that in 5 out of the 6 targets, our probes show a wider dynamic range where you can see more data points are amplified. And so why better dynamic range is important, you might ask. In simple terms, this means that they allow the detection of targets that have different quantities in the sample. So this will ensure that you can get more accurate result with your samples. For multiplexing, adding more targets to a single reaction can lead to competitive inhibition between the different targets. So this can result in change in performance. The change in performance is observed as a shift in CT when you compare the reaction run with single target versus a reaction run in multiplex reaction. So to illustrate this, we have the Tacman QSY2 probes compared to a supplier probe in a 6-plex reaction. So on the top, we have the difference in CT, so difference in performance for the multiplex reaction versus the single-plex reaction for either us or the supplier. So a greater bar indicates more change in performance. As you can see with the Tacman QSY2 probes in red, there's a minimal, if any, change in performance. And this is obvious as you can look at the raw CT value for the same data on the bottom, where orange is the multiplex CT for the target, and then yellow is the singleplex CT for that particular target. So for all the different targets, you can see that our QSY2 probes show minimal change in performance, if any. And all of these indicates that, using probes that are specifically designed for multiplexing can help ensure the accuracy of your results, as well as consistency in the performance. Choosing the right assay for multiplexing is crucial, but it is just one part of the equation. Another really important factor is choosing a master mix that is suitable for your multiplexing application. So at Thermo Fisher, we have 4 different GPR mixes that are tailored to different application needs and validated for multiplexing. So all of them are also designed with inhibitor tolerance in mind in case that you encounter difficult samples, which can further reduce the sensitivity on top of, scaling up from 1-plex to multiplex reaction. Today, we will focus on the TacPath Duraplex 1-step RT qPCR master mix. This master mix is new on the market and it takes RNA as template, and it is specifically designed for higher order multiplexing, allowing up to 6 targets in a single reaction. So this can be a very powerful tool for multiplexing, as you will see in the data we have after this slide. So TacPath Duraplex 1-step RT qPCR mix allows you to amplify up to 6 targets in a single reaction. And aside from this key feature, it also have improved handling, in-use stability, and robust to freeze-thaw. So all of these are important for having a master mix that is reliable and stable. And that's not all. So this master mix can also be used to amplify sample that have inhibitors. And finally, it also has reduced viscosity, as well as a full workday assembled reaction benchtop stability that can allow it to be used with liquid handler and automation in high throughput screening. So why Duraplex? Why use it for multiplexing? As we've touched on, a broad dynamic range is crucial for the accurate and precise detection of all targets in a multiplex reaction. And that's where TaqPath Duraplex one-step RT-qPCR master mix shines. It has a impressive dynamic range in multiplex reactions. To give you an example, here we have a 6-plex reaction with sample input of 9 different dilution points in a tenfold dilution series. And as you can see, for all 6 targets, you get evenly and consistent amplification curves with the different replicates. And with this wide dynamic range, there is less concern about losing detection in targets of low abundance in a multiplex scenario. Efficiency and linearity in amplification are critical for all targets in a multiplex reaction. However, these can be compromised when you scale up from single target to multiplex reaction. So to illustrate this, we want to see how Duraplex compare to supplier 1 and 2 in amplifying a 6-target reaction. So here we have 6 human gene expression targets with varying sample inputs in tenfold dilution series. So the log10 RNA of input is plotted against CT and fitted with linear regression to get the R-squared value that indicates amplification efficiency. When using a reagent that is optimized for only up to 3 targets, such as in the case of supplier 2, we observed a large reduction in sensitivity where only 2 to 4 dilution points can be detected per target. Furthermore, the consistency between targets are also less, as you can see in the gray shaded areas for the 95 confidence level, it's wider for some of these targets. And in the case of Duraplex, it's able to amplify all 6 targets with precision and accuracy as indicated in the narrow gray regions and the data points being very close together. And it also has a wide dynamic range for all 6 targets shown here. In other words, using a master mix designed for higher order multiplexing like Duraplex can help ensure reliable results in your multiplex RT-qPCR reactions. So for multiplexing in qPCR, we've talked about how competitive inhibition can occur between the different targets, and that scenario can be especially bad if you have something of high copy number next to something with low copy number. So to illustrate how Duraplex handled this, we conducted a study where we used a fixed input of internal positive control xeno-RNA at 10,000 copies per reaction. So here it is seen with the SINE 5.5 channel. So this fixed input is used against a dilution series of 7 dilution points of human gene expression targets. And as you can see, the amplification of the Xeno is consistent regardless of whether or not it's with low input from the human RNA side versus the high input in the human RNA side. And vice versa, the amplification of the other four targets remain consistent regardless of whether or not it is with the xenoRNA at either a low or a high input. So you can see the R-square value remain very close to 1. So this implies that Duraplex can confidently amplify samples of varying input with high efficiency for all targets. And then on the right here, we have the amplification curve for the same 5-plex reaction. So this, we want to show that we ran this with a supplier real-time instrument as an exercise where we check the dye compatibility first with this instrument. So here we use Fan Vic June, standing 5 and standing 5.5. We do know that the June is detected with the ROX channel here, so we can tick that when we use the supplier instrument. So this way we ensure that all the dyes we use in the multiplex reaction is compatible with the real-time PCR instrument here. So another common concern to multiplexing is the change in performance when you scale up from single target to multiple targets in a reaction, as we show in the QSY2 probe data. So here we're also showing that, but in this case, we're using the Duraplex master mix. So Duraplex master mix in a multiplex reaction is graphed in red for each of the target, and that of the single target reactions is graphed in blue. So as you can see that all differences across the different targets are minimal, if any. When graphing the multiplex reaction CT difference with the single target reaction CT, you can see that the changes are all below 0.3. Therefore, all in all, we can say that there is no change in performance when scaling up from 1-plex to multiplex reaction using Duraplex. So one challenge that customers or us might face is the reduction in sensitivity when you combine different targets in a single reaction due to competitive inhibition. This situation can worsen when you deal with difficult samples where PCR inhibitors further reduce the sensitivity. To demonstrate how Duraplex handle this, we ran reactions with and without an inhibitor and compare the difference in CQ for each target by the reagent supplier and plot that as delta CT on these graphs here. So on the left, we use hematin, an inhibitor commonly found in blood samples. And just as of a note, this concentration used was quite high. So we expect to see delayed CQ for reactions with this inhibitor. So the higher the bar indicates the more change in performance with the inhibitor reaction versus no inhibitor control reaction. So comparing all the different suppliers, you can see that supplier R and Q have the worst performance when it comes to this multiplex reaction performance with our Orphate 1AB engine, S-gene, and Xeno target. And our TACPath Duraplex in no rocks and rocks in red and white red stripe are the best in performance in terms of multiplexing when encountering inhibitor. So we also see a similar trend with the spiked in 50% viral transport medium in multiplex reaction. We use the same 4-plex viral assay with an IPC and saw that, supplier QB and supplier Q did the worst, and TACPath Duraplex in red and white red stripe showed the least change in performance between the inhibitor reaction and no inhibitor reaction. So these indicate that Duraplex performs robustly even when faced with inhibitors in a multiplex context. So reproducibility is the cornerstone of any research or diagnostic work. So here we've independently manufactured 3 lots of Duraplex one-step master mix and examined their performance in multiplex reactions. What did we find? Consistency across the 3 different lots. On the left, you'll see the no rocks version of the master mix, which demonstrated consistent performance across the 3 different lots as indicated side by side in a 6-plex reaction with the different colors for the different targets. And on the right, you'll see the version of this master mix with rocks in the 5-plex reaction. Again, the different lots side by side with different colors indicating different targets. Both of these graphs show no change actually in performance between the different lots. So all in all, this reagent designed for multiplexing is also very consistent lot to lot, and this can help ensure that you get reproducible results in your multiplexing reactions. In conclusion, multiplexing and qPCR require many different considerations to ensure reliable and accurate results. Assay design plays a particular crucial role in the overall outcome with factors that can impact the reliability of the results. However, with the right tools and resources, such as the Tacman QSY2 probes and Duraplex one-step master mix, multiplexing can be made significantly more manageable and efficient. These products offer superior performance in terms of sensitivity, dynamic range, and efficiency when you scale up from single to multiple target reactions. Altogether, while multiplexing in qPCR has its own set of challenges, with the right tools at your disposal, you are better equipped to navigate these complexities and harness the full potential of multiplexing in qPCR in order to increase the productivity and reduce the time needed in your workflow. As a reminder, the QSY and QSY2 probes that we highlighted today for the excellent multiplexing capability are available for order through our custom assays on our website, and you can feel free to explore these resources at your convenience for enhancing your multiplex work. And as today we've discussed the particular impressive performance of the Duraplex master mix in the multiplexing reactions, it's worth noting that there are many other features of this product that makes it even better as a tool for your research. It has features such as extended stability at room temp and refrigerated conditions, compatibility with automation with reduced viscosity, and increased benchtop stability in the assembled reaction, tolerance to inhibitors, a little bit of which we covered today with the multiplex context, as well as it being very reproducible lot to lot. So all of these features will make it a valuable addition to molecular diagnostic testing applications, as well as in R&D applications for the biopharma sectors. So feel free to explore how this tool can enhance your work if you need. Again, I have links to the multiplex PCR guideline for Tagman chemistry that's available online. So that link can be provided with your request. Similarly, with the links to the pre-designed assay search wizard, as well as the custom assay design hub, where you can design your assay with self-service tools or request help for helping you multiplex for your specific needs. And finally, I also provided links to the TACPAP Duraplex one-step master mix if you're interested. Thank you for your time today, and I will take any questions.
Dr. Yan, thank you so much for this informative presentation, just fully packed with info on successful multiplexing and qPCR. So we're going to dive right into our live Q&A. As a reminder to our audience how to submit questions, just type them into that ask a question box, click submit, and we will answer as many of your questions as we have time for. We already have a ton of questions coming in, but before we start, I do want to bring your attention to the far left of your screen. You're going to find a lot of helpful resources, and a little further left of that is a survey that we ask you to fill out, and your participation is greatly appreciated. Okay, Dr. Yan, I'm just going to dive in here. We'll start at the top and work our way down. Our first question, how can I deal with low quantity genetic material samples?
So right off the bat, because we know it's low in quantity, right? So you would ideally use a master mix that's perhaps more concentrated that can allow you to have a higher sample input, so then you can maximize the amount you put in the reaction in order to get amplification. And the other obviously the other method would be try to re-extract this sample, right, if possible. I wouldn't recommend going straight away to using a purifying, concentrating type of column unless you do have a large volume but just low concentration. Then go ahead and do that, so you know the exchange, you'll end up with a lot less volume because you do lose a little bit in this type of purification column kits. So yeah.
Thank you. Our next question, what constituents of a master mix make it more optimal for multiplexing?
Yeah, so there are, many things in there that helps it be good at multiplexing, and it's a really complex system, right? But generally, it is in terms of the characteristic, the things that's in it that makes it good for multiplexing will allow it to have a wide dynamic range and have good sensitivity. So the wide dynamic range is its ability to pick up, targets of different levels of expression or abundance. And obviously you want sensitivity, right? So you may lose sensitivity when you put different assays together and the reagents are used for different targets. And specificity is of course another thing. But yeah, there are a bunch of different things in there that can help with that.
Thank you. Now, our next question gives a little background before the question, so bear with me for a moment. Hi there. I'm trying to target two different alleles that are very similar in my multiplexed qPCR, and I'm having problems with specificity with my probes. The probes are amplifying on the on-target and off-target allele. However, the off-target allele has 4 to 5 CT difference between the on-target. Sorry. Aside from redesigning the probes, can you recommend any other parameters that we could optimize to increase the specificity of the probes?
Okay, so with the info that I'm given, so first thing, yeah, of course, the probe redesign is that something that's probably obvious, because that's depending on the length of probe that can um, affect its specificity, as well as depending on the two probes, like I don't know how close the TMs are. If they're a little different, you could change the thermocycling conditions and try to see if you can increase the specificity, say by increasing the annealing temperature. And then something else you could play with is perhaps changing the concentration of the probes. And again, without more info, this type of question, I would probably need more info, but always make sure you have positive and negative controls to check the probe performance. Thanks.
Great, thank you. And to our attendee who submitted this question, please know that the questions can be submitted during the on-demand period as well. So if we only answered part of your question, feel free to submit a more detailed question and we can help you there as well. Let's see, our next one. What are the differences in MGB and QSAY, QSY, and QSY2 probes? Is it the type of quencher?
Yes, that's right. So there are two different type of quenchers that is recommended for the Fenwick Abijun dye. So the difference between them is the MGB will have the minor groove binding moiety that increases the TM, so it will allow you to have shorter probe sequences. That's why if you switch between the two, you will need to redesign that. And with these two quencher choice, again, depending on what you're trying to do, you may want one over the other. And there's some information online, and we're here to help you if you need.
Excellent. Our next question. Are real-time instruments preset to capture all reporter dyes for multiplexing?
So yeah, that's a good question. So some real-time instruments will have the excitation emission filters turned on for all the channels, but some won't. So you really want to double check, aside from dye compatibility of your real-time instrument, you want to make sure before you run the experiment, the excitation emission filter that you are using for your multiplex reaction is actually turned on. And that's not a silly mistake, because it can happen day to day for us too, right? Like we're rushing and then we forget to turn it on. Thank you.
Great. Now we have time for just a few more questions. What can you do if the expected expression level of your different targets are very different in a multiplex reaction?
So you can definitely play around with the assay part. So you can try changing the amount of the primers and probes for the different targets. You can limit the amount of the primary probe concentration that you put in for something that's super high that you're worried it might be out competing the low abundance target.
Great. And Dr. Yan, we have time for one more question. I know you can see these questions as well. Which one would you like to end with?
Let's see. I can take that one.
Okay. So Dr. Yan, I'm a clinical bacteriologist. Often during early infection phase, bacterial load are very low. I always worry that our IAC, the human gap DH, may overcompete bacterial pathogen DNA. How should we validate that false negative is not happening?
So this you would definitely play with your assay design, right? So this, if you're worried about that, I would definitely put in lower amount of the assay targeting the human target to make sure it's not happening and that you're able to amplify. And I guess the best way to go about testing that, I would probably have sample that are of known quantity. So like, the amount of the human RNA or DNA that you're putting in, and the known bacteria DNA amount that you're putting in, and titrate and then see if that over competition is happening before you go into like real samples. And again, I don't know if there's bandwidth for you to test that, but I would do that before testing real samples.
Thank you. And Dr. Yan, thank you for your time today and for your important contributions in successful multiplexing and qPCR. Do you have any final comments for our audience?
Yeah, so I appreciate all of you for attending this, and I really hope that some of you can have some takeaway about multiplexing, because it is a complex, it's a complex process to get to the optimal results. And again, we have a lot of great resources on our website that's free for you to explore, and I would encourage, if you have questions, try to search for them in our Thermo Fisher website, or request the links here. So I linked some resources for you. And again, thank you all for your time.
Dr. Chia-An, thank you again. And we do want to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. I do want to thank our audience as well for their interesting questions. Additionally, today's webcast can be viewed on demand, and it will be, we'll send you an email when it's available for replay. So we encourage you to share that email with your colleagues who may have missed today's live event. And if you missed filling out that survey, it will pop up on your screen in just a moment. So to all our friends out there, thanks for joining us. Have a beautiful day. Take care, everyone. Bye-bye.
Thank you. Take care.